Hello, what we're going to do today is we are going to finish off C4 and just begin D4. Just touch on that. Alright, so that's what we're going to do. But first I want to go through this picture, because I promised you I would give you an update on my garden. And this picture was taken in the evening when there's a lot of shadows, so you can't see it as well. But what I want to focus on is this peony. Now, in case you heard me saying this word and you know what I was talking about, this is what I was saying. This is this type of flower. And they're really, really beautiful flowers, so peony. Now, when I first took the first video, you might not even see these, otherwise they might have been maybe about this tall. That's it. And this is just about 3 weeks later. And they've grown on. Now look, there's actually a peony. You can't see it very well here, but yeah, it's really tall. It's about, compared to this one, it's like, give you a different color. It's like up here. Oh, not there. It's like up here, this tall. That's a lot taller. So there's a lot of growth, a lot of photosynthesis taking place in my garden. Alright, and you might wonder why all these are reddish. Because they have a pigment. Let me get a different color here. Maybe black would work. They have a pigment called, let me write it here for you. A-N, A-N-T-H, I don't know if you can see that very well, O, C, Y, A, N, I, N, S. Anthocyanins. That's that red pigment. Remember, there's a lot of accessory pigments. So this is anthocyanins, and these plants have more of these than the chlorophyll. It does have chlorophyll, and it is using the chlorophyll, the green pigments, for photosynthesis. But it has more of these reddish, purplish pigments than chlorophyll. Okay, so let's get started. Let's go to C4 and finish that one off. What we're going to be looking at today are changes in chromosome number. So we're going to be looking at abnormalities in chromosome number and also in structure. We're going to talk about monoploidies, polyploidies. Remember, ploid referring to set. Monoploidy, one set. Poly, more than two. So there is a change in the number of complete sets of chromosomes, and we have talked about this several times. And it is lethal for most animal species, and plants actually can tolerate it a lot better. Let's talk about monoploidy. So if an organism is usually diploid, a monoploid only has one set, mono one. It occurs rarely in many organisms. Monoploids do not survive. However, certain species have monoploid organisms as a normal part of their life cycle. Like, we're going to look at bees. Let's go ahead and look at bees. The gender in honeybees is not determined by sex chromosomes. It's actually determined whether the egg is fertilized or not. So there's three kinds of bees. We have worker bees, queen bees, and drones. So the queen, basically all her job is to lay eggs. And she mates with about 15 to 25 drones, which are males. And then she just stores all the sperm. So she could store like thousands and thousands of sperm, and she will mate with maybe 15 to 25 drones from different hives. And so, let's hear. These are bees. These are bees. No, these are bees. It looks different here. Bees. So in this case, if it's fertilized, it will become a female. And that's a symbol for female. And it will be diploid. If it is not fertilized, I'll just keep the same color. Yeah. It is not. So if it is not fertilized, it is going to be haploid. Right now, color. So the ones that are fertilized, the females, they are workers. There's the workers. And this fertilized egg actually will become either a worker or a queen, depending on what the workers feed it. This one right here, this will become a drone. So the workers, if there's like a lot of honeybees in their hive, what they will do is they will feed one of the growing eggs, fertilized eggs. They will feed it something called queen jelly or royal jelly, and then that will become a queen. If they feed it worker jelly, then it will become a worker. The reason why sometimes if there's a lot of honeybees, they will give the fertilized egg the royal jelly because they want her to go and take a lot of bees with her and form their own hive. So here they are, they're feeding this one. If this was a royal jelly, then it will become a queen. Not royal jelly. If it is fed worker jelly, it becomes a worker. And these ones are with stingers. They can actually live about more than 2 years, but these only going to live about 3 to 6 weeks. So just between here and here, what kind of food they get. Again, male or female, depending on this one, whether they get fertilized or not. So these ones are haploid, and these ones are diploid. Diploid are females, haploid are males. And polyploidy, poly, again, more than 2 complete sets. It's not uncommon in plants, and that's because many plants can undergo self-fertilization. So if a plant is produced with it has to be even, okay, it has with an even polyploid number of chromosomes, like 4, 6, 8, it can still produce fertile gametes and reproduce because it can reproduce with itself. And they actually have used these in the past to develop plant hybrids that are quite large, like the bananas that we have today. They actually used to be quite small, and then through actually artificially making them go through polyploidy, they made them larger. In humans, the most common type of polyploidy is triploidy, which is always lethal in humans. So some examples: polyploidy, multiple sets, chromosome monopole, only one set. Haploid means half, not one. So if normal is tetraploid, then haploid is diploid. Polyploids are common in agricultural crops, contain larger cells, larger produce, more vigorous growth, and in even numbers. Triploids are not so good, no pairing during meiosis, so they're going to be sterile. Sterility could be good. If there's something growing that you don't want to grow. So plants, remember, plants can tolerate polyploidy, but animal cells do not. Humans do not. Let's review the difference between trisomy and triploidy. Trisomy, an aneuploid that has a chromosome in triplicate, like duplicate, doublet, triplicate. Let's see if we can find which one it is. Right here. As opposed to this, every single one has 3 sets. Look at that. 3, 3, 3, 3, 3. The trisomy, remember, we're going to write it like this. Most of them are normal, well, normal number, but we have an extra one. Let's try some here. 3 of every kind, 3N. Here's some common examples of polyploid plant species. Triploid crops, bananas, apples, ginger. Tetraploid, tetra, 4. Crops, that could be a different kind of wheat. Look at different kinds: maize, cotton, potato, cabbage, wheat, tobacco, peanuts. Hexaploid, chrysanthemums, bread, wheat, oat. Octoploid, strawberries, dahlia, pepper flower, pansy, sugar cane. Polyploids with odd number of sets of chromosomes are sterile. So bananas are sterile. And they purposely make it a triploid so that they get a larger size. So polyploids can quickly produce new species. Remember we talked about speciation? Well, that's in D4, speciation. Remember this when we talk about it. Produce new species, sympatric speciation of plants that are larger and stronger than their diploid relatives. And they can purposely do this. So let's say we have a normal plant. They'll get this chemical right here. Maybe it's pronounced colchicine. Alright, they're going to apply this at the terminal bud of the branch, and that will cause it to be a polyploid. In this case, tetraploid. And that causes the leaves and the fruit to be a lot larger than the normal diploid. The cuttings of the foreign branch will produce tetraploid plants with tetraploid flowers and fruits and seeds. Actually, many of the fruit that we see in the stores that are seedless, they're because of this. Monoploid. Triploid plant lines are usually sterile and sometimes seedless. Look at our seedless watermelon, seedless grapes, seedless bananas. Actually, bananas usually have seeds in them. But because our bananas are triploids, they don't. So let's review aneuploidy. Extra or missing chromosomes, and in most cases it is going to be lethal. Again, we're seen more frequently in plants, less likely to be lethal in plants. And we did this in lab. I did a lab video on this. The causes of aneuploidy are non-disjunction, failure of homologous chromosomes to separate during meiosis 1, or a failure of daughter chromosomes to separate during meiosis 2. So if A, the failure of homologous chromosomes to separate during meiosis 1, and the homologous pairs separate, In metaphase 1, the pairs line up, and they're supposed to separate. Diploids are supposed to become haploids. The whole point of meiosis 2 was to convert the haploid dyads into haploid monads. Right, something's going to happen to one of them. So let's just review it. So non-disjunction of meiosis 1, right here. Remember, the pairs are supposed to separate. See, they're supposed to separate. See here, they separate. They separated. That's what's supposed to happen, but they both stick together. That's going to affect the rest of it. Here, the pairs separated like they're supposed to. But remember, the sister chromatids are supposed to go apart, and they don't. So that one has an extra one. So these ones, right, they're going to have 3. These ones are going to have 1. These are going to have 3. 1, now look at that. So 3, 3, 1, 1, 2, 2, 3, 1. So aneuploidies are caused by a non-disjunction in either meiosis 1 or meiosis 2. Here's some examples of autosomal aneuploidies. Make sure you know them. This actually over here is my younger sister. She has Down syndrome. And I think she's pretty cute. She's quite amazing. She is such an encourager. She's my walking buddy. Sometimes we go on 2-hour walks. And she is amazing because she is so forgiving. If someone is mean to her and you say you're sorry, it'll be like she has forgotten it. My mom taught her to play the piano. She was even playing Chopin. So Down syndrome, trisomy 21. So 3 number 21s. So something happened, non-disjunction took place. Number 21 chromosomes did not separate, and both went to one egg cell. Patel syndrome, make sure that's trisomy 13. Edwards syndrome, trisomy 18. These ones right here, the sex chromosome aneuploidies, this is a review from our lab. So you might want to review that, the human genetics lab. Female Turner syndrome, monosomy XO. This is actually one of the, pretty much the only monosomy that will survive. Poly-X, more than the normal 2. They used to think these are called super female. I don't know what that would mean, but trisomy. Males, Klinefelters, XXY. Jacob syndrome. Remember Jacob syndrome from the lab. This is the only time we can tell, number 1, who the problem is with, the dad, and we know the problem is in meiosis 2. For the other syndromes, we cannot tell for sure. So those are dealing with abnormalities in chromosome number. Now we also have some abnormalities in chromosome structure. So chromosomal abnormalities are caused not only by changes in chromosome number, but also by changes in the structure. But the key thing is the structure of one or more alleles. So we can have something called translocation. And actually there's several different kinds of Down syndrome, and translocation over here, Down syndrome, part of 14, goes down to 21. That's about 4% of all Down syndrome. So the attachment of part of one chromosome to another could be some deletions. Some genes are missing. Part of the short arm of 5 is missing. You can have certain parts duplicated, so extra copies of certain genes. Translocation Down syndrome, large arm of 21 is translated to the long arm of another chromosome, usually 14. And you might have heard of fragile sites, a place where part of a chromosome appears to be attached to the rest of the chromosome by just a thin thread of DNA. You might have heard Fragile X syndrome, Martin-Bell syndrome, defective allele at the tips of both chromatids of the X chromosome. Right there. Here's the normal one. And then we have something called genomic imprinting, where certain genes are expressed differently depending on whether they're inherited from a paternal or maternal parent. So you can determine whether inheritance is from the mother, the male or the female. And here's the examples that I have for you. We have two different disorders, Prader-Willi syndrome and Angelman syndrome, and they're both due to deletion of the same part of chromosome 15. However, they actually produce two different syndromes, though you might expect them to be the same. Prader-Willi if it's inherited from the father, Angelman if it is inherited from the mother. And we can see they're actually quite different syndromes. Again, they're caused by the same deletion on chromosome 15. If the problem is from the father, Prader-Willi syndrome, small hands and feet, short stature, huge appetite, mental retardation. If the problem is from the mom, uncontrolled muscle movement, feeding issues, so they're not very good eaters, so that's quite the opposite. Very happy to meet her, laugh a lot, unusual seizures, and mental retardation. And then we have diseases that are caused by a single gene mutation. We have two different kinds, autosomal recessive and autosomal dominant. So let's look at the autosomal recessive disorders first. Again, we went through this in your lab, in your human genetics lab. I did a video on it. So many human genetic diseases are autosomal recessive, and therefore they're going to express only in the homozygous state. There's some examples: PKU, sickle cell anemia, cystic fibrosis, Tay-Sachs disease. You do not need to know these ones. The only ones you need to know are the ones we talked about in an actual lecture, which are sickle cell anemia and albinism. Albinism and sickle cell anemia. And autosomal dominant disorders, Huntington's disease or Huntington's chorea. We talked about this is one that can be hidden and protected, so the effects are not going to be expressed till 40 or older. They do have a test for it, but no way to cure it. And let me review this. So natural selection acts on the phenotype, though the genotypes are passed on. So I did this in the lab video. Dominant mutations are not usually seen because they're always expressed and they're usually lethal. The only dominant mutation disease that we're talking about is Huntington's disease. They're going to be normal to around middle age. It's a neurologic disorder, progressive deterioration of the brain, no effective treatment. However, it's on a timer. The person who has it may not even know they have it, and they have passed on their DNA. We talked about recessive mutations can be hidden. They can be protected in the heterozygote condition. Therefore, recessive mutations can be carried forever in the population. And they're going to remain hidden. They're going to stay in the population until two heterozygotes mate. When they do a heterozygous cross, 25% chance the offspring with the recessive condition will be born. And this is one reason why inbreeding is not very good, because it brings out all the bad genes. So more of a chance that we'll have two of those recessive mutations coming together. Let's start on D4. And I was going to do all the genetic problems on this video, but I think I'll just do those on a separate video so you'll have all the genetic problems together. So macro versus micro. Macro means big, micro means small. So for something to evolve, that means something is going to be growing, developing, like languages can evolve, evolution of the airplane, getting more and more advanced. Exploration of spaces, evolving, evolution of decades of research. Biology, change in the gene pool, of a population. Gene pool is all the DNA, all the genes in that population. So a change in the gene pool of a population from generation to generation by such processes as mutation, natural selection, genetic drift, and a few others that we'll talk about it in D4 and also Lab 12. Make sure you watch the videos. So I'm going to do D4 and Lab 12 together. And then I'm going to do a separate video of Lab 12 with all the math. So if you were asked to write a definition of evolution, you can say it is a genetic change in a population. Microevolution, small changes. You may not even be able to see them. Macro, when we talk about macroevolution, we're going to be talking about speciation. Alright, that's what we're going to be focusing on, speciation. When we talk about Hardy-Weinberg, when we talk about microevolution, this is also called population genetics. And you know, in biology, we like to have a lot of names for the same thing, population genetics. And we're going to be looking at Hardy-Weinberg principle, or equilibrium. Equilibrium, or you can say Hardy-Weinberg principle. So these are going to be small changes. These are going to be bigger changes. So we have one species, something happens, could be they are separated, or maybe polyploidy takes place, so they can't interbreed with each other anymore. So they'll become two different species. So we're going to start out with the micro. We're going to be looking at changes in gene pool frequencies. So we're going to be looking at allele frequencies. And first we're going to try to figure out how can we keep allele frequencies the same from generation to generation. How can we keep the allele frequency stable, that means the same, without changing from one generation to another? And that's what we're going to be learning about next. Alright, until then, have a great day, have a great study, have a great study.